Hi, I'm Zach March, and I want to introduce our recent review in brain research highlighting diverse roles of prion-like domains as epigenetic regulators, scaffolds for subcellular organization, and drivers of neurodegenerative disease. So, what are prions? Prions are infectious proteins that can adopt many functionally distinct conformations, at least one of which is self-replicating. In yeast, several proteins are known to form prions, including SUP35, URI2, and MOT3. These proteins all have a domain composed of only a few amino acids, particularly uncharged polar residues. This means that these domains are predicted to be intrinsically disordered in solution. And it turns out that these domains are essential for the protein to undergo the conformational conversion to the prion form. Interestingly, the exact amino acid sequence of the prion domain can be scrambled and still encode prion behavior. Thus, it is the overall composition of the domain and not the precise amino acid sequence that encodes prion behavior. So what do prions do? Most yeast prions bind nucleic acids and have roles in gene regulation. A classic example is SUP35, which is a translation termination factor in yeast, so when it isn't folded into a prion, it recognizes nonsense codons on mRNA to stop translation. On the other hand, when the majority of SUP35 molecules within a cell are sequestered into prion fibers, ribosomes read through stop codons and translate into the 3' untranslated region. It turns out that a wide variety of stresses can cause SUP35 prion formation. And these environmental stresses are just the sort of thing that necessitate increasing genetic heterogeneity within a population of yeast to increase the population's environmental fitness. So, SUP35 prionogenesis acts to unleash cryptic genetic variation in yeast to increase the fitness of yeast in response to diverse stresses. Another yeast prion, MOT3, is a transcriptional repressor of Flow11. MOT3 prions have recently been shown to be environmentally responsive modulators of multicellular growth phenotypes in yeast undergoing fermentation. When yeast begin producing ethanol by fermentation, MOT3 represses Flow11. However, ethanol accumulation stresses the yeast and triggers MOT3 prionogenesis, which results in loss of Flow11 repressor activity and drives yeast into a multicellular growth program that results in increased metabolic efficiency in what is now a low glucose and hypoxic environment. Do human proteins have prion-like domains? We recently applied a prion-like domain detection algorithm to the human genome and detected 250 proteins that harbor a prion-like domain. Remarkably, the gene ontology terms for these genes with prion-like domains shows that they are enriched for proteins that bind nucleic acids and have roles in gene regulation, which is a fascinating parallel with the roles that yeast prions have in gene regulation in yeast. Moreover, several of these, including TDP43, FUS, ataxin-2, and TAF15, have connections with neurodegenerative disease. These diseases are characterized by protein mislocalization and accumulation in insoluble aggregates within the cytoplasm of neurons. Therefore, we suggest that human proteins bearing prion-like domains should be scrutinized as potentially causative agents of various degenerative disorders. However, there are 250 prion-like domain-containing proteins, and so presumably prion-like domains serve some beneficial or essential function. Recent reports have illuminated what this normal functioning might be by suggesting that prion-like domains may facilitate critical phase transitioning events that provide organizational scaffolds for membraneless organelles, such as RNP granules and nuclear subcompartment. For example, FUS forms dynamic assemblies that display physical characteristics of liquid droplets. That is, they display fast internal dynamics, as you can see from the FRAP experiment at the left. If you photo-bleach one half of a FUS droplet, you see that the FUS molecules from the unbleached half of the droplet rapidly relocate to the bleached half, leading to a rapid fluorescence recovery in the bleached half of the droplet. Additionally, the droplets have roughly spherical morphology and fuse rapidly when brought into close proximity with one another. So these studies have enlightened a new model to understand how prion-like domains function in normal biology and how dysfunction can lead to disease. In this model, proteins with prion-like domains can reversibly phase transition into liquid droplets, and this process may underlie the formation of diverse membraneless cellular compartments including stress granules, P-bodies, and nucleoli. However, improper persistence of stress granules can lead to their maturation to a more solid and irreversible form, which is pathogenic and may form the basis for the insoluble protein plaques seen in other degenerative diseases. Although these current reports focused on phase transitions of prion-like domains, which have very distinct primary structure, it will be interesting to see in future studies whether or not other intrinsically disordered proteins occupy similar phases.